Still getting people. All right, I guess we can go ahead and start. If you haven't already, there's a link in the chat to, actually, does the chat show up for people who are coming in? Like, do you guys know what I'm saying?
The chat is there, but I believe that if you joined after you posted the links, then they won't be in chat.
Right, okay, I'll just post them again then. There's a lot of good links I'm posting right now. But yeah, so if you haven't already, do the attendance form. Sign up for our newsletter if you aren't already. Can everybody hear me okay? Yeah, all right. Cool. So I don't have too many announcements for CACSCB stuff right now, but there are some opportunities that I want to point out. These links I also put in the chat. But tomorrow at 9 a.m., there's a SOUL seminar with Dr. Chris Schnell from University of Washington. He's a urban ecology professor. Shell. Oh, did I spell it wrong? Oops. Shell, not Schnell.
I would highly recommend. He's a great speaker.
Yeah,
an awesome researcher. I do a little bit of work with him, kind of.
Oh, really?
A little bit. He's part of the same network.
Right. Yeah, so I posted the Zoom link. That's tomorrow at 9 a.m. And then there's also on Thursday a Chalk Talk. I don't know too much about that. Jeff, do you know what that's all about?
I actually just asked my roommates this, or other grad students. The Chalk Talk's more like more of an interactive seminar where it's for when they're hiring people or they're trying to recruit people and everything. That's like the chance to get to ask questions, sort of almost like interview kind of questions, but it's like anyone can ask them. It's like what, just to like get their more direct research ideas and things like that. And, I'm curious what that's going to be like.
Yeah, I unfortunately can't make that one, but I'm going to be looking into getting recordings and sending those out to people. I can't make it. But that's the Chalk Talks on Thursday. The seminars tomorrow both at 9 a.m. I also posted a link for Soul's undergraduate research on Handshake. They have 2 opportunities right now that are pretty nice. One's the Urban Bird Physiology Behavior Lab. And then there's another one with like ant physiology. So that's pretty cool. I'd look into that if you are looking for a lab. And then also if you're graduating soon and looking for jobs, the Phoenix Zoo has some jobs opening up. I just found out about this today, but like there's a conservation technician position and then 2 keeper positions. I believe it was ectotherms and elephants. So yeah, that's exciting stuff. Those don't open up too often. So if you can and you want to, like, apply for those for sure. But that's all that I have for now. Yeah, Jeff, you want to take it away?
Sure. Am I able to share screens? Yeah. No, it's disabled.
No? I can.
Go away, fly.
I don't know how to do this.
I think that'd be down in, you should have a thing that says like security, probably.
Oh,
right. The host.
Right.
Yeah.
Oh, you should be able to.
Can I now? Oh yeah, now I can. Okay, cool. So yeah, I threw together some slides kind of haphazardly, which I can share these at the end of this too. Actually, maybe if I just share that screen. There we go. Cool. Everybody see that? All right. Hi, everybody. My name is Jeff Haight. I'm a PhD student here at Arizona State University. Some of you I recognize from Bio 322. So good to see you here right after class. So yeah, today I'll be talking, Zach asked me to come give a little bit of a workshop / introduction to camera trapping and how we use it, what we do, how we use it for wildlife research. So just a little bit more general info about myself. I did my undergraduate at UC Davis, studying environmental science. Then I took a few years off and just did some field research kind of things, get some experience. Thinking I would study sort of landscape scale processes and how wildlife move across landscapes. And that led me to this master's at Utah State. But really what I was most interested in studying is wildlife in cities, because I grew up in a city where in the Bay Area in California, and one of my impressions was always like, how do animals actually get around here? Because you see all these stories of wildlife wandering into the city, and I was just baffled me. I'm like, how are they persisting here? So that led me here to study my that is sort of the driving research question for my research here at ASU, which I do that with under the advisement, or whatever the word is for that, of Dr. Sharon Hall and Dr. Jesse Lewis. So I'm kind of split between two campuses. So yeah, many of you either have or probably have had Con Bio with Dr. Hall. So camera traps. If you're completely unfamiliar, sorry, is there too much wind coming through my microphone? It is pretty breezy here. Okay, good.
I think you're good. There's a little bit of wind, but it's not too bad, at least for me.
Cool. So yeah, what is a camera trap? In case you're completely unfamiliar, it is essentially a remotely triggered camera, such as this one or the one pictured, which this might be the same one. I don't know. They come in a different varieties. These bugs are distracting too. Why did I choose to sit outside? I don't know. So some of them have what are called passive sensors. So they are triggered by movement, and in particular, a warm moving thing is what actually triggers the camera to take a photo. There are some, especially, not older ones, and then the other variety is ones with active triggers where they shoot out like a little laser beam, like a tripwire. And if something passes through it, that triggers the camera to take a photo. But these have their history. They got their start among hunters, really, so they could keep track of game and the usage of these different game trails and everything. But pretty quickly, that was also right around the time when conservation biology was becoming a thing. So pretty quickly, conservation biologists and ecologists realized you could use it as a relatively low cost. It's gotten a lot cheaper, but definitely a more less invasive way of studying wildlife, especially these wildlife that you don't necessarily see if you go out on a given day and look with your eyes. So it has gained traction. They've gotten a lot cheaper. On the lower end, there's ones that are like $100, $200 or less each, but they can go up to, if any of you seen like Planet Earth 2, or I think Planet Earth 1 also, there's just footage of using these like $1 million HD camera traps, essentially getting these high resolution photos of and videos of tigers and snow leopards and animals like that. So there's a range, but most often what you see being used for research are these more affordable ones because scientists were broke. Yeah. There are certain drawbacks to wildlife cameras as a sampling method. First is that for most species, you only get what's called detection, non-detection data. You only have like, you saw the species or not. You can't really tell how many individuals were there in a given amount of time unless it's a species with distinct markings or you give them tags. Also, they don't they're not very effective for detecting certain organisms, especially reptiles, since they're not warm-blooded, and small mammals, just because they're so small, it's hard for them to trigger the cameras. So they have their limitations, but they can get a lot of good data. And when it comes to setting up a whole, I should have put a map in here of our study areas, but when it comes to setting up a project to look at wildlife population or community in a given area, there's lots of considerations, of course. There's a lot of rigor that goes into this, into determining what is the appropriate number of cameras, how long do you need to keep them up, how do you space them about, how do you take into account variation in certain habitat or potential habitat characteristics, such as chose these photos at the bottom to represent the a gradient of urbanization that we're using for our study here in the Phoenix Valley, going from, we wanted to set up the cameras to capture that gradient from least urban on the left to most urban on the right, which that's actually downtown ASU campus. So lots of considerations in the design of the study, but then there's a whole element of sort of what you could call expert opinion, but it's that, like, once you have your sites picked out, you kind of have to just do what you think is going to be most appropriate for maximizing the probability of actually detecting the species you're going to detect. So say you had to set up a camera in downtown Phoenix, where is the most likely place that you're going to find something in downtown Phoenix? So there is a level of thinking like the animal, I would say, which is kind of the fun part, I think, but the challenging part in setting up the cameras at the sites. So I'm going to attempt this. Zach asked that I try to do a little demonstration of how to set up a camera. That's part of why I'm in my backyard right now. So I'm going to see how this goes. I guess I'll stop sharing here so you can see my screen a little more, maybe. So this is one of the types of cameras that we were using for our study. This is a Bushnell Trophy Cam HD. Only like $100. I kind of want to get one for myself if I had money to throw around, just to see what I have when out camping and stuff like that. But they're relatively affordable. First thing you would do is you put batteries in, put the card in, memory cards, because I would say a typical, at least at our sites, a typical month for one camera is probably going to be, on average, like 500 photos-ish. It varies quite a bit, but you can choose your settings. Probably hard to, I don't know, probably not too easy to see. These batteries are almost dead. But you can choose your settings. You set the clock so that, like, each photo will get a timestamp that will help you estimate activity patterns and things like that. So there's lots of settings you can do. You can change. Oh, this camera's dead. Okay, that's all I can show on that. I think this was a broken camera in the first place, potentially. So yeah. Anyway, you can change things like the number of photos that it takes every time it's triggered. So we have it set to take 2 photos every time, so that you have a better chance of actually, like, getting a good photo of what it is you're trying to ID. You can change the rest period between how often it can be triggered. We had it set to be like every 30 seconds it would be triggered, and you could that's all I have to say on that. Settings vary, especially depending on how expensive you want to get. These ones just have an infrared flash, so the night photos are all black and white. But if you get slightly more expensive ones, you can get ones with color flash at night, so you can get clearer night photos. These do take video, but that takes up a lot of data, essentially. So we weren't really doing that. I forgot to say, if there's any questions at any point of this, just speak up. Let me know.
Can you talk about
Yeah.
the trade-offs between like infrared flash versus color flash when they're used, but then also like the photos versus video?
Not sure what trade-off it is, sort of. Well, video, I would say the main trade-off between photos and video is it takes a lot more time to go through videos, even more so than it does to go through photos, because you're gonna fill up your memory cards really quickly with data, because the memory cards we were using could store like 50,000 photos, but not that many hours of video. But where that becomes a little more useful is with video, you can start to look at things like behavior. I know there's a study that I think Dr. Shell was involved in that's been going on across North America, where they're looking at how coyotes, how the boldness of coyotes changes within the city, like in relation to urbanization. So they were setting up these bait stations and these like novel objects, and they would videotape the coyotes so that they could actually look how they're interacting with these novel objects. And then again, really windy. Sorry. So yeah, that's one of the advantages to video, is if it's a good way if you want to look at things like behavior. And but it takes up a lot more time and data. Then infrared and color flash, I would say that's roughly just money because the color flash ones just are more expensive in general. But infrared is usually fine for actually getting, for being able to identify animals.
Hey, Jeff.
Yeah. Mm-hmm.
Sorry to interrupt. I didn't know. Are you looking for specific animals, or are you doing like an overall survey of what you find in the cities?
Yeah. So I'll talk a little bit about my project, and I'll share some photos later. But my dissertation is focusing on the whole community, you could say, like basically everything it can find. Most often, camera trap studies are really, your study design is going to be pretty specific to a species. Like when it comes to picking how you space out your survey locations and everything like that, you want that to be based on the home range of the species that you're choosing. How long you keep the camera out, like how long you make your survey last, will depend on like what species you're trying to detect. So you see some, like quite a few projects like us who are trying to look at the whole community, but ultimately you're designing these with a particular species in mind. And I think the protocol that we're based off is essentially coyotes, but we're using it. It's still appropriate to use it for all the species. Sorry, that was a long-winded answer. So yeah, I could show how to actually set this up. So this is my backyard. Let's say I have actually seen a gray fox run across that back wall. So I'm curious to see, so getting to your site, one of the first things that you're essentially going to be looking for is what's like the most likely path of movement? Because remember, we're trying to maximize the probability of detecting something. So I don't know, there's no good obvious wildlife corridor. I haven't seen like any tracks or anything along the base of that wall, but I think if a fox is going through my yard, it's probably going to be on that back wall. So I'm going to try to aim it at this back wall because I think that'll be the most likely corridor of movement. Now, the other consideration is like, what is the what's the best tree or whatever to attach it to. Like I thought about using that big mesquite over there, but it's kind of far away and doesn't really actually get like the main part of the wall here. This tree actually isn't very good because it's kind of small and it's gonna rock around a lot and get triggered falsely. But just for demonstration purposes, pretend this is a larger, more sturdy tree. So when setting it up, we have our sort of path of movement, what we predict to be the best path of movement, at least, right over there. We want the camera, in order to maximize the chance of this actually being triggered, we want to be as like perpendicular as possible to that path of movement, because just the way perspective works, any movement along that corridor is it's going to appear as a larger amount of movement than if we were like lined up with it, like something walking up and down it won't really, if you're walking towards the camera, it doesn't actually look like much side-to-side movement. So we want to be as perpendicular as possible. So I think this is my tree. Oh, I didn't, let me grab something. Typically, you want to protect your cameras. So like this camera is only like $100, but you want to make sure that your stuff doesn't get stolen or knocked off by a bear or something. So most of them, most cameras you can buy these lock boxes for, which they're not crowbar proof, not hacksaw proof, but they prevent most cases of vandalism and stealing of cameras, which happens even out in the middle of nowhere. So normally we would screw this onto the tree with like some spacers, like some washers and stuff to sort of adjust its angle. But I'm not going to screw the box into this tiny palo verde, especially since that takes way too long. So I'm just going to use the strap that comes with it. So I'm going to put it in here. No. It says it does allow for a pretty easy setup. Go bouncing around. So the most common placement is about knee height. You'll see it in papers. They'll say like, oh, between so-and-so and so many centimeters. Really, just everyone sets them at about knee height is good, because too high up, I don't know, that just seems to get like a good enough field of vision. So when you see an image from a camera trap, like the sensor only goes up to like half of the photo, sort of. So only like the bottom half of the view will actually trigger the sensor, at least with these ones. So one of the things we want to do is make sure we get the angle right as well. So knee height is pretty much standard throughout camera trap research. But we also want to make sure that we get a good angle on it. So currently, that is not pointing down at the ground really. So we're going to actually want to do something to change that angle, like put some little stuff in here like this bark. That should adjust the angle enough, I think. So attach it to my tree. If I was using the box, I would, like I said, I would have like some spacers, like some washers and nuts, things like that, metal hardware to sort of adjust it. And it's tedious, takes some time. But what I want to do as I'm adjusting it is I want to get that angle right where I'll kind of like stand back here and just like look at the top of the camera and see like what what's it lined up with. It's kind of just stand in front of it, try to get out. Yeah, try to get a feel for where it's pointing. And I think that's probably a good angle. Maybe could tilt it down a bit more. But what you will also want to do is check, do some test photos. So I've set it up, but I would want to, like, I don't know, set my water bottle or something like that here, and just like walk by it a couple of distances so that I know that it's actually being triggered. You can probably see the red light flashing. It seems like it's being triggered nonstop, which is probably indicative of this camera being broken. But yeah, you can see when it's being triggered. You go and take the card out and sort of see what the photos look like, adjust as needed until you get it just right to where you've got a good angle, like you're, so that anything walking by in the spots you are thinking things are going to walk by is actually triggering the camera. So yeah, adjust as needed, make whatever little tweaks you need. That's pretty much it. Any thoughts on that? Questions?
Yeah, I have a question.
Yeah.
So, yeah, so like for studies, you have this like large amount of data and all these like pictures. To sort through it, do you ever use any like programs or anything to sort it, or is it all just by hand?
Perfect. Great question. I didn't see who that was. But yeah, great question. I will actually continue on with the rest of the slides I have because that was the next thing I'm going to talk about. I'll try to go a little quicker because I was worried that I wasn't going to be able to fill time because I'm like an hour. I haven't put together an hour presentation like forever. So yeah, that's actually the next thing I was going to talk about. So we've gotten in the field. The next step is going out and checking the camera, which usually just involves making sure the area in front of it is clear, weeding, typically trim branches that are in the way, swap out the memory card, retrieve your data, swap out batteries. That's the field work. But you're retrieving all these photos, you're getting, I don't know, depending on the length of project and whatever, you could have anywhere from tens of thousands to millions of photos to go through. So we've got to sort through those and tag them all. So in one form or another, there are different sort of protocols and platforms for sorting through photos. We use this database that was made by our partners at the Urban Wildlife Information Network. We upload all our photos to there. They have these little things with drop-down menus where we pick what species is there and things like how many there are. We can highlight them. And then when we click next, save and next, it'll go to another photo. And we repeat the process. You can see this set of photos is 3,441 photos. This is Pima Wash in South Mountain, if you're familiar. So there's lots of recreationists and lots of wildlife. But yeah, this is one platform that is kind of exclusive to our research network, but it's based off of similar, more widely available platforms. Some people are part of a group called eMammal and use their website, which I'll mention that a little bit more later. But this is how most camera trap photo sorting is done, essentially, is by hand, clicking through. Some people just use their have file folders and just like move them into different folders based on the species and things like that. That's another method. But essentially you have to go through them one by one. Sometimes you see there's like a batch tag thing. You can sort through a bunch of blank photos at once, for instance. there's things to make it go a little quicker, but it's going through it manually, which can be tedious, but kind of rewarding when you come across something cool. Now, I think, did I throw this in here? Yeah, so that is how most of it's been done for a long while, but these days there's a lot of work that's being done with machine learning and artificial intelligence to automatically sort through camera photos. Because as I mentioned, some of these projects, including our own, have millions of photos, especially with these false negatives where like vegetation is just setting off the camera. So there's these sort of alternative efforts going on. One is this new one called Wildlife Insights, which is in association with Google. And then Email, which is a platform that has been used for a while. They're starting to do work with Microsoft in their AI for Earth initiative. I'll share these. Yeah, we'll make sure to share these slides in case you want to use any of these links. Yeah, so there's work that's being done if you don't want to go through thousands and thousands of photos tediously, but you're really into coding and programming and big data things like machine learning, then there's a space in wildlife camera trapping. So this is a hot topic that is getting a lot of attention these days. So then once you have all your tags on your species, you have like just these long lists of photos with a time and a species and a location based on your site. Sometimes you keep information about the number of individuals in a given photo. But really, this is the simplest form that the data comes in. Like now it's not raw data anymore. Now it's actually data we can use for analysis. So for instance, like on the left, you have your raw data sheet where it's just like I said, site, time, species, number, site location, exactly. So with data in this form, you can, since you have times associated with it, you can sort of filter out different species and just use that. You can filter it out by seasons, like time periods, but you can estimate things like activity patterns. On the right is this from a paper looking at Amur tigers, where they estimate based on camera photos, they estimate the sort of diurnal activity patterns of tigers and other species, including their prey species and species they're interacting with like humans. And you can sort of look at the overlap between the activity, the fine scale activity of these different species to potentially look at how interactions among species are changing with time, or how those species are changing their activities in different areas, like if you're near development or far away from development, if you're near a water source or not near a water source or something like that. You can look at how activity patterns differ. Another common way of using this data, though, is to look at patterns of, you could call it presence-absence data, but really all the zeros that you see are not actually absence. It just means an animal wasn't detected. So you split your whole study period into these different survey occasions, these different sampling periods. This could be like a week, it could be a month, depending on how long it is, depending on your species, how frequently things are detected. There's different lengths it could be, but you split your whole study into these different time periods. Say this is week 1, week 2, week 3, week 4, week 5, or occasion 1, 2, 3, 4, 5. And then it's just, was the species detected at that site or not? And this is coyotes across some of our study areas. And from that, you can estimate one of the things with this data, though, is that the zeros, like I mentioned, aren't really true absences. It just means the species wasn't detected. But what that means is we can actually predict, based on off of these patterns, since we've sampled on multiple occasions, we can predict the probability of detecting that species at a given site. And from that, we can predict how many of those zeros were actually the species being present. So you can account for what's called imperfect detection. And yeah, anyway, as a long-winded way of saying, you can, through these like multiple surveys, like replicated surveys across space and time, look at presence absence data and estimate the use, or like the likelihood of use of these different sites across something like a urbanization gradient, for instance, is what we're doing. So this is, I forgot to label it, but this is coyote occupancy. So it's the probability that a coyote is present at a given level of urban land cover. Anyway, we can see it decreases. So another use for it. Okay. There's a lot of people doing camera trap research at ASU. I don't want to pretend I'm the only one. It's just I'm only really qualified to talk about what we're doing with our study. But I do want to mention that there are some real top researchers, in particular Jorge Ahumada, who works for Conservation International, but is based at ASU as well. He's very big in this field. Jan Skipper from the Phoenix Zoo, and et cetera. But so there's lots of people. I wasn't able to list them all here, but yeah. Our project is run through the Central Arizona Phoenix Long-Term Ecological Research Site and partnered with the Urban Wildlife Information Network. So this is the aerial view of the Phoenix metro area. This is our study region for our project. This is the study area of CAP-LTER. Yeah, so across this, ooh, the lag in these is, so across this whole study area, we wanted to look at wildlife populations and communities in relation to urbanization. So we looked at this like proportion of land, urban land cover across the valley, and then we set up sets of cameras in these different levels of urbanization. So there's some in the most wild areas, like the Estrella Mountains to the south, the McDowells to the east, and the White Tanks to the west. Then everything, then all the way to downtown Phoenix, like the ASU downtown campus and next to Sky Harbor Airport, and then everything in between. So this big stratified random sampling design so we could look at urbanization's effect on wildlife communities. So finally, the good part. I'm sorry there weren't more pictures of cool animals before. That's, like anyone who does wildlife camera research, it's like there's a photo of a cute animal every slide. I did not do that, unfortunately. But anyway, these are some of the most common species that we see. Coyotes, bobcat, gray fox, javelina, cottontails, jackrabbits. To sort of orient you for people who are familiar with the areas, that coyotes just in east or sort of North Mesa near one of the canals. Both those rabbit photos are from the ASU West campus. Next to the dorms, there's that big solar field. If any of you have been out there, we had a camera out there for a while. That bobcat is at Shaw Butte, sort of in the middle of the city, but one of those sort of islands of habitat, good habitat. And that gray fox is actually on the Tempe campus. If you've ever seen the gray foxes around the Tempe campus or just around Tempe, there seem to be a lot of gray foxes around Tempe, even in the most urban areas. It's like right next to Frat Row, pretty much. Yeah. And the javelina's up in Cape Buttes, I think. Other more common species, we do get lots of photos of certain ground-dwelling birds. I mean, a camera is not great for birds because most of them fly, but the ground-dwelling species, it kind of works. Like we get lots of photos of quail and roadrunner. But unfortunately, they're not really being included in many of my analyses because I'm looking at the mammals. And I wish someone would do something with it specific to the birds. Anyway, since our project is an urban study, we also get lots of photos of dogs, cats, peoples, and horses. We have a camera that, or had a camera that was out at the Tonto River, so we got lots of photos of, Tonto National Forest, the Salt River. So we got lots of photos of the wild horses out there. And then lots of photos of people riding horses in more urban areas. And lots of photos of people. Can't really share those for obvious reasons, but I can share a photo of myself. Tried to perfect my camera trap selfies over the years. Some less common species, here are a few. We do have lots of kit fox at certain sites. I could have also put mule deer in here also. We've only gotten a few photos of mountain lions because we're not really in these like higher elevation, more mountainous areas. We do get badgers, but not too many. And most excitingly, last week we found our first river otter, which was weird, which I don't know if many people know that there are otters in the Verde River in the Salt River. They were reintroduced back in the 80s, and they made their way over to the Salt River at least. So we got that little cutie last week. Yeah. All right, so then we also do have certain species that are pretty hard to tell apart. Zach asked me that I talk a little bit about some of these species that are hard to identify. The skunks are probably some of the harder ones, I would say. From looking at this, how many species of skunk do we think we see here?
4.
4?
I was
That's
also gonna say 4.
4. So actually, it is just 3. All of those ones on the left are the hooded skunk. Hooded skunks have quite a variation in their color patterns, as you can see, but one of the things that sort of unites them is this massive wispy tail that usually is like up on high. But they go from like completely black to mostly white on top and sort of everything in between. Then we have our striped skunk, which is in more urban areas, the most familiar to everyone probably, with its usually 2 white stripes down the back, smaller tail. Then we haven't had a lot of photos of hognose skunks, but we do have one right there. They're all white on the back, but smaller tails. If you get it, can't really see it in there, but you can see its snout is a little not as hairy. So it's sort of like a bear snout. It's how you tell it apart. But yeah, our skunks are pretty tricky. Our foxes are pretty tricky, especially at certain sites and at certain times of year, because especially in the night photos too, because you don't really see much of the coloration that's on the gray foxes, because kit foxes, you can see on the left, are pretty evenly beige all over. Although those sort of, the 2 on the right of the kit fox, I'm still not 100% sure to this day. They may be gray foxes. It's hard to tell. But some of the things that help you tell them apart is the kit foxes are pretty evenly colored beige, and they don't have, you can see in that far bottom right one, the gray fox has that like black stripe that goes up the middle of its back. They're also a lot grayer, obviously. They sort of have like a 2-tone color too, where their underside is whiter. And then their proportions are quite different. You see the gray fox have a lot longer, larger tails, sort of like a feather shape. But yeah, they can be tricky.
There's a question in the chat.
Yeah, oh yeah, sorry.
Okay, it says, okay, he asked you this a while ago, but
Oh,
bring it
sorry.
up now. How far is the range of detection for the camera?
That is a good question. I want to say depends. No, an ecologist's favorite answer. No, 30 feet, I would say. I think that's a typical range for most of these cameras. At least for these cameras, I can say. That's like the max range for detection. It also, 30 feet for detecting something like a coyote or a fox maybe, not necessarily for detecting a kangaroo rat. So it does, like, they give a number for what the range is, but it does depend on the size of the thing that's moving too, and how hot it is. So yeah, it depends on those other factors, but 30 feet is typically the range. So yeah, you don't want to choose a tree that's too far back from what the path of movement you think is going to be. And I am going to hurry up a little bit. This is just to say there are plenty of photos, especially these night photos with the infrared flash, that are pretty tricky sometimes. That upper left is a jackrabbit, but it's a kind of small jackrabbit. And you kind of have to go off of things like its posture and the relative size of its ears, but it's kind of blurry. Sometimes something like a javelina in the upper right will be real up close, but some things that can, how you could tell that's a javelina essentially is because of, I don't know, its shape, its butt, and the hair actually. There aren't many species that have really coarse hair. But then there are some really tricky ones like this lower left, that coyote there. It's pretty blurry. I think Zach actually ID'd this one too, incidentally, might have. But we both seem to think it was a coyote. I should say we usually have multiple people sort through these photos so that if one person's not so sure and another person is, then there's like a step of validation to really be extra sure of what the ID was. And we both agree that this is a coyote, as blurry as it is. And then mule deer can be pretty, or just deer in general can be pretty tricky to ID. And I don't know if I really have time to go into telling the difference between the different deer species right now. But most of what we see are mule deer on our cameras. You can tell mainly here by the tail. Yeah, so what to do if you want to get involved with these kinds of projects. First, I would say that there's a number of sort of citizen science programs to give opportunities for just like members of the public to get involved, especially through sites like eMammal. And also a lot of our partners at the Urban Wildlife Information Network, which I should mention is a network of a couple dozen cities throughout the US that are doing urban wildlife surveys, following a similar protocol so that we could look at how wildlife respond to urbanization and live in cities, et cetera. So I check them out, especially if you're interested in urban ecology. A number of those partners do like have just members of the public help them sort photos. And also just reach out to people like myself and other researchers who you hear are doing wildlife camera kind of work. Also reach out to me if I don't have, I would say currently I feel like I have my hands full in managing people, but we always need, like, anyone who's doing wildlife camera work actively needs help sorting through photos at least. And these days it's kind of hard to go out in the field to get like hands-on experience. But one of the advantages to photo sorting is you can do it remotely usually. So we're keeping on working through the pandemic. But if I'm not able to provide that opportunity, feel free to reach out to me and I could try to connect you with someone who could use the help. But I also do need help. I think that's all Any questions on camera stuff? No. Anybody muted? These last few minutes, I did want to put in this little plug for SEEDS, which is a program of the Ecological Society of America. I'm not sure who's been around for it, but there was a Seeds chapter at ASU, and I've spoken with Zach a little bit about this, how it would be cool to try to get it going, to give students at ASU another opportunity to connect with another professional society with which a lot of you will probably end up working. I know that your group is already associate. You are the chapter for the Society for Conservation Biology. This is sort of an equivalent program for the Ecological Society, you could say. But more specifically, it's to promote ecology opportunities for underrepresented students, to create inclusive environments within the field of ecology, offer these like hands-on engaging experiences, connect diverse groups of ecologists and students. But yeah, so this program, so if you have a chapter or if you're associated with one of these Seeds chapters, there are lots of opportunities you could get access to, like undergraduate research fellowships. They do field trips both regionally and nationally that you can go out and network and learn a lot of things. They help with attendance at conferences, in particular the ESA annual meeting. They do these leadership meetings. If you're listed as a chapter, you can even get money, I think, to go on field trips and things like that. And yeah, it's a great way to sort of connect into this as a student, really like find your way as an ecologist. I would say it was a very instrumental program in me becoming the ecologist that I'm becoming, or whatever. I was involved as an undergraduate, much as a lot of you are involved in CACSCB and Nature at ASU. So I've just wanted to put that little plug in that if people are interested in sort of restarting the ASU Seeds Devils thing, I would be glad to sort of give some guidance on that. It's a very minimal effort thing where it's like you submit a report every year and you have an advisor listed. You wouldn't even have to do much, but you would be connected to all those sort of opportunities. You could probably I don't want to mess with the whole ecosystem of environmental clubs at ASU, but I don't want students to miss out on opportunities. So I just wanted to bring that up. And I think that's all I have.